Hi everyone. My name's Gary. I'm excited to present some work that I've done jointly with Uyghur Mai, my advisor Ben Raphael, on inferring the history of cell divisions and migrations. So as we all know, cells constantly divide and migrate. This happens at very small time scales, such as injury response, but also at large time scales, such as organogenesis. The key question then is, given only observations at the present time, can we infer, one, how our cells ancestrally relate to one another, but also two, something interesting about the spatiotemporal process that describes the locations of related cells. There's a new technology that might help us answer this question called spatial lineage tracing. This figure that I'm showing on the screen now comes from It Memoir, which was published in Science 2020, and it shows how one can combine the traditional dynamic lineage tracing system, where we insert genetic barcodes into the genome of a progenitor cell that we can then induce inheritable and irreversible mutations at. What's new about this technology is that we can also now record the spatial locations of those cells at the time of sampling. To be a bit more formal, we're given two things from these experiments. One, the sequence mutation information, which we call this character matrix S, which is a cells by sites matrix, where the entry is the mutation state of a particular cell at a particular site. Furthermore, we also have these 2D locations for each cell, which we denote as matrix L. Thus, our problem is, given the character matrix S, the locations L, and a tree topology T, we want to find branch lengths denoted by these deltas and other model parameters that maximize some likelihood that I'll talk about soon. Those of you that are familiar with cell lineage tracing know this is part of a broader problem where it has seen a recent surge of methods. And so all these methods attempt to find either a topology T and or branch lengths for each edge of that tree, where these branch lengths represent scaled real time, and these internal nodes represent division events. And thus, given data only at the leaves of this tree for your last final sampling, we want to infer the topology and or the branch lengths. And as I mentioned before, there are a large number of existing methods that try to solve this problem. However, none of those aforementioned methods attempt to model the second form of information, these 2D locations, and thus new methods are necessary. I say new methods a bit willingly because actually this is quite an analogous problem to species phylogeny or a subproblem in species phylogeny known as phylogeography. And here we're given sequences and spatial information for perhaps or ranges of locations for species. We want to see something interesting about the ancestral patterns of those ancestral species, as well as model a tree for those species as well. And thus, our contributions are twofold. We define a novel spatial process for the locations of related cells, and we incorporate this spatial model with a sequence mutation model to improve estimation of branch lengths given the topology. So as a first stab, one might look to Brownian motion. This is the most widely used spatial model, not only in the cell migration literature, but also in phylogeography. And has a very simple formulation where if you're given the location of the parent cell and you're interested in modeling the location of a child cell, you can typically take the parent cell location and add a normal random variable to each of the x and y coordinates. This random variable will have variance with a sigma parameter, which is a spatial diffusion rate, also scaled by the amount of time since division, or in other words, the branch length. So a natural question when presented with this Brownian motion model is that it's mathematically valid, but does it actually describe the movement of dividing cells? And to answer this question, we can turn to video microscopy data. And so luckily for us, It Memoir, one of these spatial lineage tracing technologies, provides some validation of their method using video microscopy. And we can turn to this data set to try to answer our question about if dividing cells behave in a Brownian motion manner. And so in their experimental setup, have mouse embryonic cells dividing on a dish for 36 hours while tracking each cell's identity and location. And so of the many statistics we can compute on this video microscopy data, one that we're interested in is this distribution of daughter cell locations. And so in particular, we're interested in this angle formed by the parent cell U at the time of division and the cells V and W, its two daughter cells, at the next time point. And so if you imagine Brownian motion, this angle of this distribution of these angles should be uniform, or you expect it to be if you truly believe in Brownian motion. However, when we actually analyze this data, it turns out that data distribution looks something like this. It's widely skewed towards, or it's skewed toward wider angles rather than being uniform like you'd expect for Brownian motion. And so it might take a few seconds to realize this, but actually cell division is a spatially symmetric process. And so this distribution is not surprising at all. And we can see that perhaps the division process is causing the skew in the angle by examining this distribution of the angles as time increases. And so we're recording the location of parent cell U at the time of division, but then we're increasing the amount of time before we sample its two daughter cells V and W. And we're seeing how that distribution of angles changes. What we can see is that as we increase this time, this distribution of the angles flattens towards this uniform distribution as you increase the number of frames or amount of time from division to sampling. Thus, this motivates this new model that we call the symmetric displacement model. We define two new terms, let R be the radius of the parent cell, and let this theta parameter be the axis of division. And thus, you can formulate these two new generating equations, which are actually quite simple to understand. And that you look at the first and last terms, it's just Brownian motion in its original form, and these middle terms represent symmetric displacement. And so what's symmetric about this is that for one child cell, V, these terms will be added to its coordinates, and for the other daughter cell W, it'll have these terms subtracted. And thus, you still retain this symmetric displacement for a given pair of two daughter cells that have the same parent. So there's one last step we need to use to actually be able to use this model for phylogenetic estimation, and that's that we're interested in a tree, not just pairs of cells. And so if we assume the x and y are independent for simplicity, let this vector L sub x be the observed x coordinates. The probability of observing the set of x coordinates is actually a multivariate normal distribution. If we assume a fixed constant r, then we can actually just calculate the mean of each leaf node by summing the displacements along the path from the root of this tree down to the leaf, plus the location of the x root. And the covariance of this multivariate normal is for between any two leaf nodes is equal to the sigma squared parameter times the amount of time that these two leaf nodes have co-evolved together. And conveniently for us, we can use a modified version of the Eisenstein's algorithm for Brownian motion to marginalize out this x root. So that gives us our model, which we call mollusk, maximum likelihood estimation of lineage and location using single-cell spatial lineage tracing data. It takes in this character matrix S, as well as the 2D coordinates for the cells L, as well as a tree topology, and performs a maximum likelihood estimation over both a sequence likelihood and a spatial likelihood to give us a spatial diffusion rate estimate as well as branch lengths. For the sequence likelihood, we actually use a pre-existing work called LAML, which was published in Recom earlier this year. And for our spatial likelihood, we use our model, spatial displacement, the symmetric displacement model. So now I'll briefly go over a few results. So to go back to this in our data set, we're interested in two metrics, the spatial diffusion rate sigma, as well as branch length estimates. And we can use data from video microscopy once again to compute true tree topologies, branch lengths, as well as an estimate of a true sigma value. Furthermore, we assume that this average cell radius is given in pre-known, and we compute this from these video frames to be around 6.868 like normalized pixel units. So let's first talk about sigma estimation. I promised at the beginning of this talk that I would say something interesting about the spatial temporal process that governs these cells. And so when we've interpreting this sigma parameter is that larger sigma parameters equates to a larger expected square displacement, and smaller ones, a smaller square displacement. And so on this plot here, I'm showing on the y-axis the bias of the sigma estimate from our method to the true sigma found from the video microscopy data. On this x-axis, I'm showing different parameterizations of our model where we can either vary this radius amount to show how changing it from the true radius amount might change the estimates of our sigma, as well as the effect of using only the location likelihood or the sequence plus spatial likelihood in our model. And so to break down this plot a little bit further, observe that when you have r equals 0, this actually becomes normal Brownian motion since your displacement terms all are 0. And in this case, it makes sense that your sigma hat estimation is greatly overestimated. This makes intuitive sense since if you're not accounting for this displacement, then you have more space that Brownian motion has to account for. Furthermore, we also tried to see what would happen if we vary this r parameter, this radius parameter, and we saw that letting it equal close to its true value led to a more unbiased estimate of this sigma parameter. And that gives some confidence that our model is actually capturing some true biology and it's not just some random coincidence that we observe lower sigmas. And finally, we observed that we're actually robust to different radius amounts and that any reasonable choice for this radius amount seems to give you a better estimate of sigma than the original Brownian motion version. Finally, I'll conclude on a branch length estimation result where this metric that we're interested in is this mean absolute percentage error. And I'm writing this on the left for a semi-simulated data set where we simulate mutation data, keep the real locations, as well as on the right for the original MMR data. And we can see that in both cases, actually, we achieve a minimum branch length error at this true radius amount, which also gives us some confidence about the biological validity of our model. So in conclusion, we introduced this symmetric displacement model, a novel spatial model for cell migration. We provide an implementation for joint optimization for sequence and spatial data. And we demonstrate superior performance and benchmarks for estimating our two quantities of interest, these branch lengths and the sigma parameter. So I'll just conclude with some acknowledgements. I'd like to thank my lab and my co-first author, Uyen, as well as Princeton and some funding from the Lineage. Sorry, say that again?
You assume that cells are independent of each other.
Could people asking questions use the microphone for online?
You assume that each cell divides independently of the others.
That's right.
Can you comment about how important is this oversimplified assumption? Because sometimes a cell can divide and other cell would disturb it.
That's right.
So you assume something that is a bit unrealistic.
That's correct, yeah.
Cells evolve independently. So
that's correct.
If you can comment about it.
You're correct that that's a simplifying assumption we make. It's something that we considered the effect of, but to the best of our knowledge, there wasn't like a lot of tractable models to try to consider how different cells might affect the division events of other cells, and also in what manner, in a more biologically realistic manner. And so that's some ongoing considerations that we're thinking about. But at the present time, we don't have like a good mathematical formulation of how we might cleanly do that in a tractable manner.
Hi, amazing talk. I'm actually working a little bit with spatial transcriptomics and trying to integrate lineage data with spatial transcriptomics.
Sure.
And also trying to kind of look into the cell migration of cells. I want to ask a little bit of like your opinion about when you consider complex cell states and cell type dependent behaviors of cell migrations. Do you have any idea of how to like more like infer cell migration in a more biological motivated manner?
So if you knew cell states like a priori, there's some existing work in the phylogenetic literature about like varying the sigma parameter between different branches or between different like clades of your tree. And so you might want to consider something like that for your problem if you knew cell types like already.
Yeah. Thank you so much.
Yeah, I'm over here.
Sure.
So this was a really fascinating talk. And as I was sort of sitting there, I was sort of thinking about how this can be extended to a more, whole organism biological framework, because you're using 2D coordinates as opposed to 3D. And so forth. And I'm just wondering, especially since I think we have a reasonable amount of time, if you would mind just sort of maybe speculating a little about where to go to put this in a much more whole organism 3D, because this is just a great model, but a first step.
So low-hanging fruit, you might imagine that you can extend Brownian motion to three-dimensional cases as opposed to two-dimensional. But you're probably asking a bit more of a more detailed answer than that. It's hard to say without knowing what type of data you would have to try to model that with. For whole organism, Yeah, I'm curious what kind of data you would have to try to model that to begin with. I'm having trouble answering that question like sort of abstractly, since I think this model in particular was motivated by the fact that we had this video microscopy data that we could sort of like test hypotheses on in the first place. And so, to the best of my knowledge, there isn't like a from embryo to, or sorry, from like single cell to small organism data set out there that's quite this resolution yet. And so, I'd be curious to see like what type of data that might look like, and I'd have to answer it given that data.
And I think that's fair. I didn't mean to sort of, push that way. Would you maybe describe just a little more about the exact data you're using to parameterize this model, even in the 2D case?
Sure.
How exactly were the data collected?
So in the IT memoir example, or the NMR video microscopy example, they have discrete time samples of this dish experiment. And so we know that for each successive video frame, that's about 15 seconds. And so that gives you, you can estimate, with the track-scale identity, what the migration rates look like if you fit that to a Brownian motion model. And then that also gives you the true topologies and like amount of times between division events, obviously, as well. Obviously, something I'll point out, or maybe emphasize, given these questions, that this data set's actually like on a dish, and so there are a lot of dynamics that might occur in a real organism that don't happen on this like dividing dish experiment, and so that's something we considered for future work.
Cool, thanks.